Hi, good afternoon, everyone, and welcome to another episode of the Botany Bistro. My name's Mary Dudley, and I'm coming at you from the Civic Garden Center in Cincinnati, Ohio. Very happy to share with you episode 15 of the Bistro. I know that we were delayed by a week, and I am fully recovered, so I want to thank you all for your well wishes. I am the Ecology Education Manager here, and I started the Botany Bistro because botany has really benefited me in my career and in my personal life as well. And I wanted to freely share the botanical information that I have learned over the years with you so that you can apply these to your gardening, your farming practices, and we can continue to raise each other up. So thank you all for joining me. If you are local to Cincinnati, we would love for you to come by the Civic Garden Center sometime and see us. And if you're not and you ever plan a visit to Cincinnati, please do stop by. But we are happy to share this virtual series with everyone who would like to be in community with us. We will be having our next live session on December 29th at noon. And so if you're interested in joining that, please visit our website and get some more details. So like I said, today we are going to be continuing our discussion on plant breeding and genetics. This is our second session for this topic. Last time we talked a little bit about the history and the basics of genetics and how that works. Today we're actually going to be talking about how you ensure that you have a genetically identical individual after you've done all that hard work of cross-pollination and getting all those characteristics correct in a plant that you find desirable. How do you make sure that you keep those characteristics going forward? And so one of those answers is vegetative propagation. So this type of propagation does not involve the crossing of any pollen. It is a clone of its parent, so it is genetically identical. So we'll be discussing some of those methods today. Whenever we have a bistro, I kind of show off what I'm having for lunch, and usually it has something to do with our topic. Today I actually have just a couple of ingredients that I haven't turned into lunch yet, but I will. And so my side dish for today is going to be some sweet potato fries. So I've got some sweet potatoes with me. I hope you're enjoying a wonderful lunch on this December day. And so we are going to talk about different types of vegetative propagation, including division, cuttings, grafting, layering. We're also going to be sharing in part 3 when we meet together next week on the 29th at noon. A little bit about how modern methods are creating resistant agricultural crops that are very important for our society moving forward. And so be sure to join us next time. We always have a little bit of homework, and so our homework for this week is going to be to try out one of these vegetative methods on your own or with a friend, if you like, and kind of share your experience with us on social media. So please do interact with us. We're here to listen and learn, and I'm very happy that we are able to enjoy our time together. So last time we talked about the basics of genetics, and when you are propagating things by seed or using pollen and you're mixing that between different plants, you're really kind of seeking a certain genetic type. You're trying to create an individual that might have a specific bloom color or bloom time or height. And so all of those things are very complicated at times, depending on the types of plant that you're using. And once you have those characteristics set, you don't necessarily want to keep experimenting, to see if it'll just continue to happen, or what if you get a different individual that doesn't have the characteristics that you'd like. So all that time and energy that you have plugged in can be replicated through vegetative propagation or asexual propagation. And so this is a different type of plant propagation that does not involve using seeds. And so we will save our seed starting tips and tricks for another day. This is talking all about propagating plants using leaves, stems, roots, different parts of the plant that we can take advantage of. Now, this is something that humans are not good at. If I cut my finger off, it's not going to grow back, unfortunately. I can't, see somebody's wonderful ear and be like, oh, I just want to swap it out and give me a different one. But plants are very adaptable in that way, and it's for a few reasons that we'll cover today. But I sometimes do wish we had the opportunities to grow as plants do, but I'm very grateful that we're able to experiment and learn from them in these other ways. So we have evidence that grafting, which is a vegetative propagation technique, has been happening for at least 4,000 years in China and Mesopotamia. And about 2,000 years ago, we have documentation of problems that these early scientists and agriculturists were dealing with when they were grafting olives and other types of fruiting trees. If you remember, some of our conversations in the past have talked about ways that growers have worked around pests and pathogen issues. And so one of those was through grafting. So most of the European wines that we have now are grafted onto American rootstocks because of that phylloxera issue that they were dealing with. And so this concept is not necessarily new. We're always developing new technologies, and we're always trying to advance the field in that way. But people have been trying their hand at this for quite a while. And so plants also do this naturally. They form tubers and bulbs, those runners and rhizomes. Sometimes those are the aggressive plants that we're a little bit angry at when they're keeping us job security in our gardens. We don't necessarily always love when our plants just decide to replicate on their own and create those little suckers and all the things that we have to manage. But at the same time, it's a natural process that they'll go through that we can take advantage of when we do want to replicate those plants and have more of them. So one of the examples that we can think about are quaking aspens, Populus tremuloides. They're actually clonal, sprouting from root suckers. And so when you see a large stand of aspen trees, all of those trunks typically belong to one individual plant. There's an integrated root system. That root system is very extensive. And when there are ideal opportunities, that root system may send up different cells to start creating a new trunk, new shoot. But that is a genetically identical individual to the other trunks around it. And I lived in Colorado for a while, beautiful botanical areas there, but the aspens were always really fascinating in the fall because they would all turn yellow, it seems, at the same time. Just big, huge masses of yellow up on the ridges in the mountains, and then they seem to all lose their leaves also at the same time. And so once I found out that they were clonal, that was a very exciting thing to think, okay, they're all operating under the same pressures, and they're all kind of working off the same material. If you don't know, in south central Utah, the largest aspen colony spans 106 acres and is considered the largest tree by weight and the largest tree by land mass. And this quaking aspen colony is lovingly called Pando, which just means I spread. And so it definitely does. It's doing a great job. I haven't been able to see this individual, but if I ever get out there, I'll be sure to see how many of those little parts of that tree I can hug. But it's a pretty amazing feat to be able to say that you span 106 acres. In non-vascular plants, such as our mosses and liverworts, vegetative structures include gemmae and spores. And in vascular plants, we're talking about roots, stems, and leaves. So when we're talking about sexual propagation, that's our flowers, and they're going to be producing seeds and fruits, things like that. But the vegetative are the parts of the plant that are not reproductive in nature. But we might be thinking, okay, how does this work? So, you can't just necessarily put like a flower petal into a pot and have that grow. So we need to know a little bit more about the science about how this works in order to take advantage of it. And so vegetative propagation is possible by meristem tissue, which is found in stems and leaves and tips of roots. These contain undifferentiated cells. So these cells have not been told very clearly what they're supposed to do yet. They're ready. They're in waiting. Am I going to become a branch? Am I going to become a root? Am I going to become a petiole or a stipule. What am I going to do? And so they're waiting for a message. And so those messages typically come from hormones that's going to tell the plant, we need this part of the plant to develop at this time. And so these cells are dividing actively by mitosis. Mitosis is the process of cell division that results in 2 copies of the same cell. Meiosis, on the other hand, is the process by which cell division happens when you are reducing the information by half that's within the cell so that it can combine with another during sexual combination. And so we call that fertilization. And so meiosis would be more for sexual propagation and breeding, whereas mitosis is the process that is just creating those genetically identical cells. And so that's what's happening in those undifferentiated meristematic tissues. Because those tissues can continually divide, plant regeneration just can happen. It's possible because we have these opportunities. Now, not every part of the plant has this meristematic tissue or has it in enough concentration that will actually be productive for us to be able to use for our vegetative experiments. And so there are 3 different areas that we'll talk about with meristematic tissue. Now, this is important to consider because areas of growth on a plant, knowing where those are is very important for pruning, for when you are taking cuttings, when you are just looking at the health of a plant. You really want to know kind of a little bit more about where these areas are. So the apical meristem are present at the tips of roots and shoots and increases the height and length of those parts of the plant. Cell divisions facilitate the growth of cells in help with cellular enlargement. So yeah, cells can be very small when they start and then they can elongate as they go out. Intercalary meristem is located at these nodes. And when you are cutting a plant or you're taking part of a plant off, you really want to pay attention to where those nodes are. A node is going to be accompanied by either a leaf scar or a petiole or a branch coming out. So if you're looking at the stem of a plant and you're seeing a branch right in that little corner, in that little nook, is what we're going to be referring to as a node. So if I'm cutting and pruning my plants, I would want to cut just below that node if I was going to save that cutting. And then if I am going to continue to have that plant in my garden, I'm also going to trim just above the next node, because otherwise the internode space doesn't have the same meristematic tissue or these same concentration of cells. And so that part of the plant is an open wound now, and it would be like leaving something unbandaged and you're just going to be like, oh, well, it'll just be okay. You're inviting opportunities for fungus, for other diseases, for pests to come in and damage the plant, because if you're cutting right around those nodes, then that action will stimulate that growth to heal that space. And so you can form a callus and you can form something much quicker than if you just leave it alone. So it's really important not to leave your internodes submerged in water when you're doing cuttings or just out left on your plants. You want to be really careful about where you're controlling that growth and where it's coming from. So that is very important for your growth. And then the third type of meristem is lateral meristem. It's located in stems and roots on the lateral sides, and it increases the thickness of the plant. Vascular cambium and cork cambium are 2 lateral meristems, and they will give rise as they divide to secondary permanent tissues. So those are 3 little kinds of types of meristem, so you know kind of where they're happening. And now we can talk about how we can take advantage of our knowledge of meristems and make sure that we are using that knowledge to create interesting new plants or clones. Let's start off by talking about probably the most common method of vegetative propagation that maybe you've tried, maybe not, but that is called a cutting. And so lots of our plants, if we're cutting them right at that node, then they have all these undifferentiated cells from their meristematic tissue, and they're, okay, all of a sudden I'm off of my plant. Which when you take a cutting, you're not usually taking a cutting of something with roots on it already. You're typically taking a cutting of a part of the shoot. And it might have, 1 or 2 leaves attached to it, but you are trying to then form the part of the plant that is necessary, which would be roots. Really, plants don't do great if they don't have a way to absorb water. and air under the soil. So trying to stimulate that part of the plant to produce roots when it has not before is a very interesting concept. We can do this by making sure that we are cutting it right at that node and putting that in either a grow medium or a cup of water and seeing if we can stimulate that root growth. You can also add a rooting hormone to this. And so I wanted to do a little bit of research about rooting hormones because I've heard things back and forth about them. And it is something extra to buy. And if you don't need to, maybe we can skip it. But it's also just, for good measure, let's just find out what it is. So a rooting hormone is a hormone that's typically in a powder form. It's like a white powder, and it has IBA, or indobutyric acid, which is a synthetic rooting chemical, and it promotes root development in a wide variety of plants without being toxic to the plant. Typically, these products will also contain some kind of fungicide, which deters damping off and rotting while the plant is trying to create these new cells and grow into a full new plant. And so what you would do if you're using a rooting hormone is you typically will take the cutting and you'll dry it off a little bit so that if there's any debris on it from other plants or anything, kind of clean up that area a little bit, just with a little bit of distilled or dechlorinated water. And then you can dip it into this rooting hormone and it will adhere to the bottom of the plant. And then you can put it into a grow medium. Now, if you're growing your cuttings in water, that isn't really going to help you much because the powder will typically just wash right off. It's replicating the auxin that would be influencing plant growth at the meristematic tissue area. And so then I'm like, okay, what are auxins? Let's go look at auxins. So these are hormones that work in tandem with gibberellins, and they increase plant length and growth. And so they are going to be, telling those cells how and when to divide. And so more of that hormone and the production of that hormone, if you can increase that production, you can have cuttings that will root faster. And so I understand why people use root hormone. Also, that fungicide is probably very valuable in areas where it might not have a lot of ventilation or just a little bit of a fail-safe. So if you want to use root hormone, please use it with caution. Follow the directions. It is a chemical, so you want to make sure you're keeping it out of reach of little ones and any pets you might have. But you can definitely give it a shot if you're interested in adding a little bit of extra boost to your cuttings. And so the gibberellins and auxins increase distance between nodes and spacing branch points further apart. These are plant growth regulators that can facilitate cell elongations and helping them grow taller. The gibberellins also play roles in germination, and fruit ripening and flowering. So it's pretty interesting. We're not going to get too deep into hormones today, but we are just going to talk about how auxins can be thought of within the plant ecosystem. So when a new shoot is growing towards the light and making sure that their roots are going down into the soil, that all depends on where auxins are located and how they're influencing the cells within the plant. The auxins move down due to gravity and laterally and away from light. And so cells will grow more in areas of the plant where the auxins are highly concentrated. And so that concentration of auxins is always kind of, okay, where do I need to go next? Where do I need to go next? How do I get closer to the light? And so it's kind of elongating that part that's further from the light so that the other parts can go closer. It's a little counterintuitive, but it seems pretty interesting to me. And it also stimulates these root branching. So when auxins apply to a cut stem, that initiates root formation at the cut. For fruit development, auxins in the fruit promote maturation of the ovary wall and full development of the fruit. These auxins are produced naturally within the plant, but like I said, we also have chemical synthesis of auxins. And these can be in high concentrations, even high enough to be considered pesticides. that can cause very drastic growth and kill plants that they just will have like uncontrollable growth. So it's an interesting balance that we're trying to search for, whether we want to kill or grow a new plant. So like I said, always read the cautions on whatever product you get. There's a few different ones out there. And yeah, to give it a try. You could try one experiment with root hormone and one without and see how different it is. And so if you're taking a leaf or a tip cutting, so this is a fun little project. And I actually have an example here of a plant that was taken from a cutting. And so we have a plant here that is a pathos. It's a pretty typical house plant. And this house plant, you can take a piece of the stem and you can chop it. And as long as you have 1 or 2 leaves above a water source and at least 1 node below that water source, there's a high probability that you will get that node under the water to start creating roots. And once those roots start to form, then they're providing what the plant needs. That plant can continue to grow new leaves and branch out. But what I'm seeing right here is a really good definition of node and internode. So we talked a little bit about how you have those intercalary meristem areas. So that's going to be here at these nodes. And then I have this internode section. And I don't have a lot of growth or undifferentiated cells happening here. So I would, if I was going to take another cutting from this plant, I would cut right here so that this node is going to be submerged. I would remove this leaf so that it's not underwater. I don't want it to rot. And then I would be able to have these 2 leaves out of the water source so that these leaves would continue to photosynthesize as the roots develop here. And then from this node, I would remove this internode so that I only have a little bit of stem happening right here so that I'm not inviting a lot of open wound opportunities for fungus and other things to attack my plants. But that's very interesting. So we've got some opportunities right here. The plant is just ready to go whenever it needs to. Oh yes, I can continue to grow and spread myself around. So pretty neat little pathos cutting. I've done cuttings with jade, with coleus. I've got some begonias that I'm doing some leaf cuttings on right now. And it's just so much fun to see what you can get to sprout just with a little snip and a little time in some water. Now you want to keep your water fresh, and I always prefer to have water that's dechlorinated so that it's not fresh from the tap. I'll put it into a cup and then I'll let it sit out for about a day to get those chlorine bubbles to gas out before I am putting my plants in or before I add it to existing cuttings. So I would encourage you to give that a try. And when you are thinking about doing a stem cutting like this, you can put it upright into a cup. A leaf cutting can be a little bit different, and that's actually when you're cutting typically right around that midrib of the leaf. You have some undifferentiated cells there, and then you are taking that part of the leaf and you are laying it down on top of a grow medium or just slightly tucked into that grow medium. And I like to use a kind of loose sterile mix. We have some seed starting mixes or some potting mixes that you can keep damp, and you can spray the leaf over time. You want to make sure that it doesn't dry out too much, but you want to give it that nice balance of just kind of moist but not soggy. And then maybe you can stimulate some interesting roots to grow from there. And so you can dip the end of that tip or leaf into a rooting hormone to speed up that development, but don't use too much. Like I said before, it can actually kill the plant if it is used in excess. And you want to put that cutting in a warm location with high humidity and bright indirect light. And so I think that you might have some success there. Cuttings that can be done in this way from stem or leaf cuttings include lots of our common favorite houseplants. We've got violets, snake plants, begonias, ivies. And so I definitely recommend that you give it a try if you have one of these plants around. Now, cuttings are not the only option for vegetative propagation. We also have grafting. Grafting is pretty Oh, I don't want to say that. I mean, it's kind of advanced. It does require some special tools, more than just a pair of scissors, versus the cutting. But if you are going to be doing some grafting, that's typically going to be done with plants that have secondary tissue. And so that's your woody stems. And so the grafting that we typically are doing now is because we want to have maybe multiple varieties of fruit or other types of edible trees together, or we're trying to protect a plant, like in the case of our European shoots of grapevines versus the American rootstock that they need to be on so that the phylloxera does not totally decimate the rootstock. And so in grafting, you're going to have a scion, which is the upper part of the cutting, attached to another part of another plant, which typically is your rootstock. So you have your scion, which is up top, and your rootstock, which is on the bottom. And you are going to create a junction between the rootstock and the scion, and there's several different ways to do that. So what you're really trying to do is you're trying to line up the vascular cambium between the two plants so that you can have a flow. The xylem and phloem needs to be restored in those plants, or else they just won't be able to survive. It would be like, cutting off your air supply. So you've really got to try to do this efficiently. I don't want to say quickly because I don't want you to rush anything, especially if you have sharp tools with you. But you want to be really prepared when you get ready to do grafting. And so there are lots of places that have kind of mother stock. And so, okay, I really enjoy this particular type of apple tree. I am going to make a lot of branch cuttings from this apple tree. But you know what? This apple tree, it just doesn't have a very strong root system. It doesn't grow in the types of soil that I want to be growing in. Okay, great. So I'm going to get another type of tree root system that will work well. And typically you are going to be staying within species for this. You can go out of species if you have the technical skill. It won't always be perfect or necessarily a quick grafting union will not always form, but I encourage you to just experiment out there. All the things that we know have happened because someone just was like, I'm just going to try it. So I hope that you're part of the try it group. And so we are thinking about how we're lining up those tissues. And so there are several different types of grafts. You can do a cleft, a bark graft, a side veneer, a splice, a whip and tongue, a saddle, a bridge, and an end arch. And there's more. But really you're typically, if we have it very simply, you would cut a diagonal cut on the rootstock and you would match that angle on your scion. And then you would put those two together and they should be the same width. The capillaries should be the same width. And then you're going to bind them together either with some kind of horticultural tape or some kind of wax, something that will keep those things together as they start to connect with each other and grow. Very cool. And so you can also try a technique called budding. And that is where you just cut one bud of a scion off and you put it into a different plant and see if it will take that way. So I highly recommend that you do a little research on grafting if you want to give that a try. It's fascinating to me to find so many different types of fruit on one particular, I don't want to say plant necessarily, but like one group. So you can have, apples and oranges and pears all on the same tree, and that they're all in the same family. They're all kind of cousins. And so it's interesting to see people's creativity. You can have several apple types on one tree. Oh, I want an orange one today, or I want a yellow one today. And they could all be right there in your backyard. So I think that that's really fascinating and just a testament to the creativity of humanity to kind of think outside the box with this. Other than grafting, we have something called layering as well. Now, layering is going to be when you're kind of inducing growth of roots and kind of independent new plants from a mother plant by stimulating a natural covering event. And so natural laying occurs when plants send out runners or plantlets. Roots will typically have already formed. So you can kind of see this in like your spider plants, when they start to kind of make babies, you've got little roots kind of on the bottom of those babies. They are ready to kind of go into another area. They're trying to get away from mama. They're trying to establish their own space. And so they're ready to do that. And so you can sever the plantlet from the parent and you can put it in another pot with some growing media. If those roots haven't quite developed, you might want to put it in the water for a little bit. But you can definitely, once those plants have formed those roots, you can cut that little spider plant off and it's its own plant. It's a genetic copy, identical clone to the mom, but it is ready to survive on its own. There are also opportunities for simple layering. And so what you can do is you can put vines on the ground that are growing, and then you would layer some soil on top. And so then that part of the vine that is submerged, you're making sure that you're hopefully close to a node, is like, oh, I'm under the soil now. I need to make roots. And so how do you stimulate that root growth? You make sure that you keep it covered. And so it understands that, oh, my undifferentiated meristematic tissue needs to now develop roots. Once those roots are developed, you can sever that from the parent plant and you have a whole new individual that you could dig up and move somewhere else, or you could keep there depending on your purposes. Compound layering is just a step up from simple layering, but it's done twice with the same shoot and produces multiple plants. And you're bending these stems into that medium. And then air layering is pretty neat. And so you're slicing a portion of a stem or a branch. And so basically you're creating a wound, and then because, you know, it's in the air, you're not able to like just bury it under the ground, you're actually going to create a little moss or soil ball around that injury that you have inflicted, and you're going to keep that covered. And so in order to keep the moisture in there, you'll typically wrap wet moss and soil around the area that you have damaged with a clean tool, nice sharp cut, and then you'll put that wet mass around, and then you'll wrap that with some kind of plastic wrap or saran wrap or something nice and tight to keep the moisture in. You will need to make sure that that stays moist and watered, but that will kind of, you know, I don't want to say trick the plant because plants are probably pretty smart, but that will stimulate that hormone to say, oh, I need to grow some roots. So those auxins and gibberellins are all active in there trying to grow some more roots. And then after, depending on the plant, after a few weeks to a few months, you could unwrap that and see roots growing, and then you could cut that plant and you could grow that in the soil. So you can create new plants just from little cuts on some of your branches, which is pretty exciting. And so other types of vegetative propagation include suckering. Suckering is when you're getting things coming up from roots. And so, like I mentioned, Pando, the quaking aspen colony, the trunks of those aspens are the result of suckering from root systems. We don't always love suckering when it happens in our yards because typically it's going to be happening around the base of trees, and they are happening in response a lot of times to damage. So if you're running over them with your lawnmower, if someone is weed whacking, or if, you know, just damage to the base of trees, that can start that kind of suckering issue. Now, I have also seen grafted plants sucker, and so you have to be careful with that because the fruit trees in my yard are grafted, and I can see the graft union very clearly, but what I want from that fruit tree is the scion, the stalk, the shoot that is going to be producing those flowers and those fruits for me. I'm not as concerned with the rootstock, and that might not be a desirable plant at all. And so if my rootstock starts to send up scions, or starts to send up suckers, I am going to want to cut those and remove those. If that is a very desirable rootstock, maybe I would save the suckers and I could plant those somewhere else to create some kind of little nursery out of those. But typically we're going to be removing suckers so that you can just keep your scion very clean. There's also tissue culture, which is taking very small amounts of tissues from plants and putting them into a container with some special medium and letting those grow into new plants. That's a really neat method that's used by people that are studying endangered species, because you don't want to take the whole plant, and you might have just a very little bit of material to work with, and so you can tissue culture from very small amounts of plants. In nature, you've seen vegetative propagation if you've been around the gardening world. Rhizomes that are growing underneath the ground are methods of this. Grasses, lilies, irises, and orchids use this method of propagation. We also eat rhizomes such as ginger and turmeric. Runners, also known as stolons, are similar to rhizomes, and they have horizontal growth just at or below the soil surface. But unlike rhizomes, they originate from existing stems. So these runners will grow and develop new roots and buds at their tips. And then you can, just like a spider plant, you can kind of separate those runners and you could cut that off and plant that plant somewhere else. Strawberries are notorious for this. And even some people I've known to call strawberries weeds because they just keep growing and going and going on their runners. Bulbs are also formed, and a lot of times these bulbs can grow and merge out. So we've got onions, garlic, shallots, hyacinths, daffodils, tulips are all examples of bulb growers. And then you have your tubers. And so I brought my sweet potatoes today. This is going to be an example of a root tuber that's been modified to store nutrients. And so these become enlarged and can give rise to a new plant. So I could actually put this in some water, maybe with a couple toothpicks to kind of hold it up so it doesn't just all sit in there. And it will start to form little shoots out of it because it's like, okay, I'll grow some leaves for you. So it's pretty neat to be able to grow a plant like that. One of my favorite ways of vegetative propagation is literally when a plant just falls off another plant. We have some kalanchoe plants around the house, and they just create plantlets. They right on the edges of the leaf, they'll make these little tiny plants that'll just fall off and they'll just start growing. And so it's pretty amazing just to see this plant that's like, all right, I'm just going to do my thing and create totally new groups of plants that are identical to itself, but they're always propagating. Like they're just never stopping. Plants are very good at just going for it. So some activities that I like to do, especially with little ones, and I don't know if you'll be visiting with little ones over the season, but you can, or you could do this with big kids too, you know, it's fun. You can create something called a root top garden. And these are going to be things that you can collect either before they go into the compost or just as you're preparing something in the kitchen, and you can grow new plants from them. And so carrots work well for this. You can also do it with radishes, beets, the tops of pineapples. I once grew an entire new pineapple plant from the cut off top of a pineapple that I had. I didn't quite get it to 3 years, which is about how long it probably would have taken to produce some kind of a fruit, but it was pretty exciting to see those new leaves start to grow. So if you're interested in trying this, a root top garden is pretty much just like what it sounds. You're cutting off the tops of those carrots. Maybe you're not feeding them to the rabbits, or you just don't have a use for them. You can put those tops into some water or a very shallow pie pan with some soil in it. Keep that moist. Keep them in contact with that growing medium, and they can start to form new leaves and new roots. And so just give that a try. Onions will also do this. You could try it with some garlic as well. And so seeing if you can propagate plants just from things that you normally would have tossed in your compost pile. So I encourage you to try some of these things. Plant propagation is always an experiment. Sometimes it works, sometimes it doesn't. But thinking about how we can go beyond the seeds that are kind of what we think of typically when we are doing plant propagation, and thinking about how we can propagate plants vegetatively is our task for the week. So I'm really excited to see you all next week. Thank you so much for joining us today on the Bistro, and I hope you all have a wonderful week.